Okay, so here are our notes for Tuesday. A lot of this is going to be review again, okay? So we're just remembering DNA. What does DNA stand for? By now you should know it's deoxyribonucleic acid, DNA. From our notes last week, we learned that there are two jobs of DNA, and the first one is DNA replication, which is where we make an exact copy. And then the other job is protein synthesis. And that is where we synthesize or make proteins. And this is the one we're going to be focusing on this week. Our central dogma is when we go from DNA to RNA, and then from RNA to proteins. This is a two-step process. So from going from DNA to RNA is step one, and then going from RNA to protein, that's step two. So here we're going to say protein synthesis has two steps. And this week we're just only going to be focusing on step one, which is DNA to RNA, and that's called transcription. Let's talk about transcription. So transcription, it takes place in the nucleus. And the first thing that happens is helicase unzips the DNA. Just like when we're doing DNA replication. Then RNA polymerase this time it is going to add RNA bases. We have our same base pairing rules, except when we're going from DNA to RNA, instead of A matching with T, A is now matching with U. So we still have C matches with G, and then we also have A matching up now with, instead of thymine, it's going to be a new one called uracil. Uracil, letter U. And then the very last thing that happens at the end of transcription, I'm going to write this again so you can see it better. It's hard to see. I don't know why the printer did this, but I even tried to make it darker. So transcription, step one, happening in the nucleus. Helicase unzips it. RNA polymerase adds RNA bases. The matching goes C to G and A to U this time. And the very last step is that the mRNA, which means messenger RNA, it leaves the nucleus. Right here, we're going to pretend that this is a little organelle inside of a cell called a nucleus. You should all know what that is. Okay. And inside the nucleus, we have DNA. And our DNA is sometimes referred to as a code. And this DNA code looks a little something like this. It's TAC, CAC, and then TAA. DNA cannot leave the nucleus, but RNA can leave the nucleus, and that's why DNA needs to be turned into RNA to leave the nucleus. Then it can be turned into a protein after that. Here's our DNA code, our template strand, and all we're going to do is our same matching that we've been doing when we make more DNA, except when we have A, we're going to match it with a U instead of a T. It'll look a little something like this. So T matches with A, and now A matches with U, C matches with G, C with G, A with U, C with G, T with A, and then U. As you can see, I kind of wrote them in a way that there's a little space in between each group of 3, and that's because there's a special name for these letters that are in groups of 3, which are called mRNA codons. They're called mRNA codons because they have the DNA code in them. And a codon is 3 bases together. So here we have 1, 2, 3 codons. So the mRNA, the last thing it does is this little strand right here. It's going to come on out and it's going to leave the nucleus. Okay, and that's going to be it for this week's notes. I just want to focus on step 1, transcription. And next week we'll do step 2, translation. That's all I want you to focus on right now for step 1. And then we'll move on to step 2 next week.